Welcome to the second how-to session of this bioinformatics workshop. If you followed the first one last week, you have seen practical tips on working with biological databases, doing blast searches and visualizing multiple sequence alignments. Today, you will have an opportunity to focus on the practicalities of using Chimera for protein modeling tasks. Further, we will take a close look at the Ensembl genome browser. Both of these tools have lots of details, but Dr. Tovanen will highlight the most useful features. Let's get started.
In our second how-to session, we'll be expanding and revising the concepts learned in the last 3 lectures, and in particular we'll be looking at aspects of data retrieval from the resources we've been seeing. And the learning goals in this session will cover a new data source, that is models created by AlphaFold, and to understand AlphaFold is really important for modern structural biology, so we'll spend 10 minutes talking about what it is, and then finally get a structure, retrieve it into Chimera, and practice some manipulation of molecular models, and also do a quick comparison of the AlphaFold model to one which is retrieved from PDB. And regarding Ensembl, we have various different ways of retrieving data, genome regions, multiple proteins and transcripts, or multiple aligned sequences from gene trees of Ensembl. Protein modeling used to be able to produce only models of quality much inferior to the experimental model, but that is no longer true. A novel method called AlphaFold has been able to bring a real revolution into structural biology, and in my opinion this is one of the most exciting things in biosciences now. So let's talk a while about AlphaFold. It is a program, artificial intelligence program, that has won the competitions of structural prediction of proteins twice in a row in the biannual contest CASP. Both contest 13 and 14 had AlphaFold as a winner. Actually, the AlphaFold of 3 years ago was quite different from the modern version, but still at both times they were clearly the winners of the overall competition, being able to produce best of the predictions. The competitions work so that they get structural models from authors who still haven't published the models, and only the sequences are given to the competitors. And once the predictions are in, they can go ahead and publish the structures, and the predictions are compared to the true structures and ranked. And even if AF1 was quite good, there were many competitors who were able to come up to its level and even beyond in the intervening 2 years. But now AF2 is really the king in the field. They were able to give the best model for more than 90% of all targets in CASP 14. And often the gap to the next best was really big, and in particular they made a huge advance in the hard targets where you didn't have any clear models that would be needed for homology-based predictions. So they were able to create good quality predictions even when nothing similar to the actual structure was known. And so all of this means a big revolution in structural biology. Basically, the protein folding problem, the task of having a 3D structure from protein sequence alone, is almost solved, meaning that AlphaFold 2 can predict models with great accuracy, comparable to experimental models for about 2/3 of all proteins, even when they are novel folds. And for the rest, most of the proteins will give pretty reasonable models. But still, the protein folding problem is not considered solved, because the kind of rules and processes in protein folding are still unknown. You can't unravel a complicated deep learning multi-layer neural network thing with millions of parameters to figure out what happens in real biological protein folding. But nevertheless, we do get a lot of excellent model. But there are some things where AlphaFold is not so good at yet. And one of them is multi-domain proteins. They can't figure out these domain-domain interactions even when the structure is or should be fairly stable. Even if the single domains are quite accurate, so the relationships between domains are not. But then there are many multi-domain proteins which do not have a regular structure, so you might have the two domains in an angle or directly in a row, so that two different crystal structures of the same multi-domain protein might show quite different conformations. And then multi-protein complexes are hard, so it's the same thing. Protein-protein interactions are not modeled as accurately as the interactions within a single domain which is folding. But then there is a new project called AlphaFold Multimer, which is able to do quite well with many protein complexes. And the biologically worrisome thing is that for flexible proteins where you might have two alternative conformations in PDB with different crystallization conditions, so you can't get more than a single conformation for any target from AlphaFold, and which conformation comes up from multiples in PDB is something they can't yet predict. And the structures when they come out are just protein, any non-protein ligands or post-translational modifications are absent. There's an estimation that more than 50% of human proteins are glycoproteins, and so missing the glyco parts is pretty serious in understanding their biological functions and pretty serious in trying to figure out interactions between glycosylated proteins. And then there is a big fraction of proteins which are unstructured. In terms of residues, there's an estimate that maybe even 30% of all of human proteome is in unstructured regions or very flexible regions, which might only take up a specific conformation in a specific interaction binding to another protein or when binding to a ligand. So we still do need experiments. The protein folding problem is not totally solved, especially not for large complexes. But with the advent of cryo-EM, or the revolution that happened there about 5 years or maybe even 10 years ago, so that we are approaching atomic resolution even for cryo-EM. So when we have these near atomic models from cryo-EM for large complexes, which is really the forte of cryo-EM, it needs large particles to be able to work. So when we have a cryo-EM rough model, docking the atomic models of AlphaFold into those complexes can give really good quality complex models too. And then what about ligand docking and drug design? That is still a problem with the theoretical models. So for the median accuracy or estimated error of atomic coordinates in AlphaFold models is said to be about 0.9 angstroms. And ligand docking needs a model or a real structure with a precision of 0.3 angstroms for atomic positions before you can do reliable predictions of a drug or a small molecule binding to a protein. And if you are looking at binding sites and active sites in enzymes, they are typically flexible, they can move, that is what allows them to bind and transform substances. So the problem with getting just one conformation is hitting especially hard in these active sites that would be important. And then another thing which is a bit hard is that membrane protein models in AlphaFold are worse than the soluble parts. This is because there is less training data available in PDB for membrane proteins, and in an opinion, scientists looked at 27 7 transmembrane helix receptors that had become available after the development of AlphaFold 2. They had a cutoff period for the training data at 2018. So there were 20 new models, and comparing the AlphaFold predictions, 10 of them were really good and 10 of them not so good. And the problem in that was that the AlphaFold parameters for their prediction reliability were not really helpful in distinguishing between the good predictions and bad predictions. So there's still room for improvement, and especially more experiments are needed for membrane proteins and their structures so that they would be better understood to create the machine learning approach models. And now we are coming to something we can use as learners and scientists, the AlphaFold structure database. In addition to AlphaFold 2 source code being freely available and the AlphaFold 2 servers being available for you to predict anything you want to do for non-commercial use. So they have made a database with models from AlphaFold 2. So the first batch that came out last year was 350,000 proteins for 21 model organism proteins. So the full human proteome except for 29 sequences has been predicted. Somebody says that maybe 1,000 of the models are not of a usable quality, but still it's pretty complete. And just very late last year, or maybe in January, they added 400,000 proteins covering the manually annotated SwissProt part of UniProt, more than doubling the size of the AlphaFold database. And their ambitious goal is to have all of UniRef 90 during this year. That would mean 130 million proteins covering everything in UniProt, so that no unmodeled protein would be less than 90% identical with something in AlphaFold database. And anyway, the models which are in AlphaFold are available both for commercial and academic use, so it is only the creation of new models which is restricted. So let's take a look at the AlphaFold database site. First, this is the list of the 21 proteomes they have. It's human and 20 other model organisms. 4 plants, a couple of prominent animal models like zebrafish, fruit fly, C. elegans, some parasites, medically important ones, some medically interesting bacteria, and an E. coli, the obvious model organism. So they have all their proteins, as far as AlphaFold could model them, presented here. And then we have the SwissProt dataset, which is another 400,000, so really mind-boggling there. Now, this is the homepage of the AlphaFold database, and we can do searches simply by keywords or by UniProt accession numbers, and there's even a link to BLAST searches when you go to, or actually FASTA searches at EBI, which you can find here in the FAQs. But if we just try our luck with keywords CA6 zebrafish, only alpha CA models from animals in PDB are from human and mouse, so zebrafish would be interesting. And we are kind of lucky. We have lots of hits because this doesn't do really an explicit and, so the best hit is the one we are looking for. And now if we look at the model itself, so this is colored by the model confidence. Dark blue colors are for very high confidence, and that is most of the properly folded domains. Then there is the signal peptide, the very beginning of the sequence. We can see that here we have with mouse over. This is hard. Leucine 4 and the turning point here is about 22, and we know that the signal peptide is the first 20, and so the signal peptide is not expected to have any specific conformation. And likewise here the end is badly predicted, and there's an alpha helical region, but it's connected by loops with unknown conformation. And they say that these regions of very low reliability are probably most often unstructured in isolation. So let's click on the download of a PDB file here, and because I've allowed Chimera to use the PDB extension in my Windows, so I'll just choose to open with, but if you don't have it, you have to choose save file and then find the file to open, but I'll just click here. The coloring by reliability, it is available there. We can go for tools, structure analysis, and render by attribute. Now, crystal structures have the B factors, which model the flexibility, the error of position for each atom and each residue. And now if we look at the properties of residues, we have the average B factor here, but now it's not B factor, but instead it's these reliability values which have been just placed in the B factor column. And in B factors, the small values are good and more rigid, so that's why we have the blue here. But now we should reverse the threshold colors, because in this reliability value scale, 100 means full certainty of the position, and the max value is 98.78, and there's a huge amount of values which are better than 90%. So let's go with this coloring and apply. And now we have pretty much the same coloring, but now we can see more precisely that the red are these poor reliabilities, and we have some unreliable loops here as well. And likewise here, the ends of the loops tend to be in the average white region of reliability. And now let's compare this with the CA6 model we have in PDB, human CA6. The PDB code is 3FE4. I fetched this like 50 times before, so we'll get it there. And now you don't see the model because it is totally outside of the screen. Now you can see it, and the reason it's so far away is that each PDB model can have its coordinate system set up as the authors like, so the models are not in the same place, not in the same orientations. And because of that, we might want to superimpose these models. Going to structure comparisons, and Matchmaker allows us to do that. And now here in Matchmaker, the reference structure is the thing we want to stay output and we choose the alpha model version here, and then the structure to match would be 3FE4, and we want to choose a specific chain B in 3FE4 because it has less. Oh, let's not. Let's just choose the best aligning chain. So we can let AlphaFold choose, and the superimpositions should be happening. All right, it's here. It happened, and we can see that we have chain A here and chain B, which was superimposed. So we can easily go and delete chain A. So using command line to select model 1 and chain A, it's here, and deleting the atoms of chain A. And this is kind of annoying. We don't need the signal peptide, so we can select model 1 residues, no, model 0 residues 1 to 20. And again, atoms bond delete, that takes out the signal peptide. And for the pentraxin domain, let's just hide it. 282 would be a good place to start the hiding, so 282 and dash without the endpoint, it goes all the way to the end. And we just go atoms bond hide, but now what we don't hide is the ribbon, so we need to go for, I'm sorry for giving the commands very quickly and not explaining them, but tilde is kind of negation of most things, so I would be doing unribbon for residues 282 and onwards. If I go with this selection and go for ribbon hide, it will hide all ribbons, and this is not what we want. So to hide just half of the ribbon, we need the command line, and with the command focus I get whatever is visible centered on the screen, and there we are. And now, as a side effect of this structure-based superimposition, we can also have a look at which parts in the sequences are easily aligned. So going for structure comparison, there is an item called match align, and now it's asking to create the alignment from superimposed models, and we'll do that. Choose OK, and there we have our structure-based alignment. So this might be of interest if you are comparing sequences in a protein family which is widely diverged, so that they are hard to align by sequence alignment methods alone. But if you align them in Chimera based on the structure, that might help you and reveal the structurally conserved regions, even if their sequence conservation is poor. But in this case it was not that bad. But now what we want to do is to study the zinc binding site. The AlphaFold model does not have any zinc there, but it probably has learned how these histidines should be placed in such a large number of CA models. And if you look at the beta sheet here, you can see the beta strand really nicely superimposed between the PDB model and the AlphaFold model for zebrafish. And now I'm going to control click on one histidine, shift control click on the other two to make a selection of of these 3 residues. And I'm just selecting atoms, so I'll go for select and broaden to select the full amino acids. Now you can see what is selected. And now if I go to the alignment, this is like magic. We have the selected histidines colored in green in the alignment as well. And now we can just mouse over the corresponding histidines in the AlphaFold model of zebrafish to find out that the histidines are numbered 113, 115. They are at the bottom right of this alignment window. And the third one is 140. And now we can select in model number 1 the residues 113, 115 and 140 in the command line here at the bottom. And sorry, I should have done that in model 0, obviously. Okay, so now you can see that the ribbon shows selection in the correct positions, but the side chains are not there. So with the 3 amino acids selected, looking at the selection inspector, I can see that 3 residues in one model. That is what we wanted. And going for atoms bonds and show, we get those histidines in place. Control clicking outside of the models hides the selection, and this is pretty good. In particular, you can see that the nitrogens which form the coordination to the zinc are really nicely in the same positions. If we had a let's have a look at how to retrieve data from Ensembl. And we will have 3 ways for doing that. First, we'll look at pages of single entries, the gene page, the transcript page, or the location page for retrieving sequences. And then we have two ways of getting multiple sequences fairly easily from Ensembl. And now, if we have just entries of one species, there's a lovely feature, Biomat, which allows retrieval, and we can even use codes from another database, human gene names or UniProt entries, etc. There are literally dozens of different options for retrieval. And finally, if our goal is to find orthologs of several species starting from a single gene, we'll look at the comparative genomics resources very briefly, just to show how we can get a few dozen orthologs retrieved in a breeze and put them into CView. And as the first hands-on topic here, we'll use a single sequence retrieval of dog CA15. Let's say we want to balance our set because there's no human CA15, we get another species of dog. And then as another example, we'll do a gene region retrieval so that we have the whole region containing the exons and introns plus 1 or 2 kilobases on both sides. So let's say we are not sure if the gene prediction is correct and we might want to use other tools for that. We won't be demoing that, but just show how to get the data from the genomic DNA. Dog CA15 gene. Let's start from Ensembl main page and select the search dog from the drop down and then CA15 as the string to search. We get multiple results, 13, and we have restrictions here. Let's click on gene so that we get 2 different genes, and the top one seems to be CA15 even if it's just labeled novel gene. And now if we want to get the protein sequence, we assume in the transcript diagrams that the very long exon is not okay, and most CAs have 8 or 9 or even 10 exons, so this looks more plausible. We take the top one as our wanted region, and then going for protein. Well, it's pretty simple. We don't want to copy paste this colored version. We want to download the sequence and amino acid sequence in FASTA format. That's okay. Go for download and opening with Notepad++. You could also choose save file, but the content will be the same. So here it is, a FASTA formatted file with the description line here and then the sequence. And now we can just go ahead and copy paste this in the end of our sequence collection to use it. The other task we had was to get the whole region of the gene, exons, introns, plus a few kilobases on both sides. And here we have it. So we have two different gene models, and we don't know if one of them might be more correct than the other. So let's do the gene prediction. We won't do it, but anyway, let's pretend we'll do that and get the coding, the whole genome region plus a few kilobases, just in case there would be additional exons on either side. So we'll go for downloading data, export data. And what we have here is a suggestion, which is exactly what we see here in the screen, but then we have to make sure we know which strand we want. Now we have the arrows pointing to the right, so this is on the plus strand, the forward strand, and there's nothing on the reverse strand. So in the export data, we have to check that we have +1, not -1 for the positive strand. I had -1 because that's what I had done on my previous search. And there's a suggested flanking region. Let's take 1500 bases on both sides, in addition to the location, which was already coming from the default of the location page, the extent of the transcript. And going for next, we want this as text, and there it is. Next we'll look at Biomart. This is a lovely data mining tool in Ensembl, and it allows us to retrieve data items from a single species, which could be even thousands. We could take, let's say, all the upstream regions of 1 kilobase for every main select transcript we find, and that would allow us to do some promoter analysis on a large scale. Or we can have just a smaller data set, like 15 human CAs, and we can do the retrieval with the help of UniProt codes. And in this exercise, we'll try to pretend that we are building a codon-based alignment, so that we need the proteins to make the alignment more reliably, and then we need the coding sequences to substitute the protein sequences with the corresponding codons. And for that, we want the sequences to have the same names and not to spend time in tedious manual editing of even 15 sequences. We'll do just 2 very quick Biomart searches, and we'll have the CDS and protein files in very little time. But we need to spend a little bit of time to show how Biomart works in general. Okay, on Ensembl main page, we can click on Biomart here or here up on the top row, and whoops, we have our previous search here, so we'll go for a new search and this is how Biomat starts usually. First we have to decide the database we look at. We are going to be genes oriented in getting the transcripts and proteins, but we also could get some variation and regulation data items in the same way. And now that we've chosen genes, next thing is to choose the species. We have every species available in Ensembl here, and we'll go for human genes. And now we have to set filters which will decide which genes we'll find, and then in the attributes selection we decide what we want to download, and then in the results we'll finally output the data we want. And now I'm clicking on filters so that we get the list here. So this would be one way of getting a specific region. So if we know which chromosome and which base region we want, so we can just get it from here without having to figure out a gene page to do that. We can retrieve data which is related to specific phenotypes, but in our case we want to go for genes and gene IDs. We can use many different IDs, so we can use the Ensembl IDs. These table IDs here are one good idea, but here we have a very long list of different things we want or which we could use, and now we want to use uniprot /swissprot IDs. And how to get them, so that's fairly simple as well. So let's jump to the list of human C8 genes which we had from uniprot. Let's go for searching and search for the greater than symbol and find all in current document. Now we have all the description lines which have the greater than symbol, 15 of them, which is the correct number. Now we can select the 15 lines, Ctrl C for copy, and let's go for, oops, we have one extra line here which we didn't want, and let's save this as ca_humannames.txt. And now we can go to Excel and open a text file. And here we selected text files, and ca_humannames.txt is here. Opening that, we need to tell that this is a delimited file, and the delimiter will be the vertical bar or pipe character. So going for delimited, that's the correct choice, and instead of tab or comma we just choose to have the tab character as the delimiter, and clicking finish. There we go. Now this is separated into different columns. We have the 15 uniprot codes here. Ctrl C for this, and going to Biomat and pasting the codes here, and let's see what we want here. Now we don't want anything at this point, we just want to do a count to see if we find actually 15 Ensembl genes. 15, that's correct. And the next thing we want to look at the attributes. Now we can select output of lots of different things. We may even get some homologs, we may get sequences, but first thing we want to build a table that gives us the Ensembl codes and the UniProt codes and gene names so that we can kind of see what we have. So gene name, and actually in external we find external references, so UniProt, SwissProt ID. So this is good enough, and now going to results, we should be getting a table. And 10 is not quite enough. We want to have them all. Okay, I'm letting the table to be loading. Oh, it's here. And it should be 15 entries. Unique results only. Come on. Let's go back and let's see if we can refine our search by adding a filter of having the main select only. So there we go, main select transcript only. So now we are not getting multiples from each transcript for each gene. And going back to results, now we should be getting a table that has just 15 lines. This is it. This is what we want. This is the gene ID, and now we only get the transcript IDs which are in the main select set. And this is good. We can save this. And as the next thing, we go for attributes again, but now we want sequences. And we have the possibility of choosing exactly anything we can get. So we can have, let's say, we only want the flanks. So this shows us the colors, what we'll get. So it is things outside of the exon. If we want to get the 5' untranslated region, it is here before the coding sequence. If we want just the coding sequence, it is here, the parts of the exon corresponding to the protein message. The cDNA has the exons so that the untranslated regions are there as well, and then we can have the peptide which also corresponds to this. So let's go for the peptide, and let's look at the header information we want to include to name all of our sequences there. So gene stable ID and version, transcript stable ID and version. And this is the peptide, so we might as well. No, sorry, we don't have peptide length, so this is good enough. Let's see what we get. And again, let's choose all. And there we go. This is 15 sequences annotated with the gene ID with the version, transcript ID and the version. Oh, this looks silly. Let's go back to attributes and header information. We want the stable ID with the version, transcript ID with the version, and now this is a little bit cleaner. Just two items here, the gene name and the transcript name. And choosing all of them, we should get the list of 15 sequences, which we can again Ctrl A, Ctrl C, and take it to the notepad and save. Note that we have the peculiarity of showing the stop codon explicitly, so this asterisk in the end of each sequence is the stop. And maybe we'll want to find and replace those asterisks, but that's another story. Depends on what we are going to use. And the next thing we want to have is the transcript sequences, the coding sequences with the same names. So we go back to results and attributes, and now instead of peptide we'll choose coding sequence. Here in what types of sequences, and we have the header information, and we want to keep it the same, and let's go for results, and again all. And as you can see, now we have longer sequences, 3 times as long to have the codons, and each sequence seems to start with ATG, the initiation codon, and each one stops with stop codons TA, ATGA, TAA, etc. And again, we can save them and use them for our codon alignment. And this works for a single species at a time or for a modest amount of orthologs. But for getting higher numbers of orthologs, we need to use the comparative genomics tools, which go across any specific taxonomic branch of species in a gene tree or some predetermined taxonomic groups which we have in the ortholog tables. So let's have a look. The gene trees, what we'll do as a hands-on is retrieval of orthologs and paralogs from a list, and then retrieving aligned sequences from a gene tree. So the task is to retrieve homologs of HDAC3. So this is the gene page of human HDAC3, and I'm clicking on orthologs here, and we get this table of orthologs. We have a summary of how many orthologs we have in different groups, and we can see that there are plenty of one-to-one orthologs, but in fish and in reptiles, or birds and reptiles, we have some, in placental mammals we have some one-to-many orthologs. So if we have duplications in one lineage of organisms, that will lead into our orthologs being one-to-many or maybe many-to-many, so that it's not a simple one instance of the same gene in each organism. But in this case, most of them seem to be one-to-one orthologs, but there are quite a few species which have no orthologs. Maybe that's a problem in the annotation, or maybe this is a dispensable gene because it comes in a gene family which has at least 12 paralogs, so this one and 11 others. So maybe the other paralogs can take up the functions of the others, so that we don't have the HDAC3 gene in exactly every species. And now if we want to download orthologs, so it takes a while before we get all 141, so I'll just choose one, let's say birds and reptiles, and download orthologs. FASTA format, we can choose whether we want aligned sequences or unaligned sequences, amino acids or DNA. Let's say we want to have alignments of amino acids, and we can choose between FASTA, Clustal W, and many other formats. but FASTA is good enough for a quick look. And downloading this, it will take not too long, and again I could save it, but now I choose to open in Notepad++ just to show what the data looks like. This is clearly aligned sequences because we have the gaps there, and we have the Ensembl protein names, and we have the species identifier repeated, and then we have the region of the protein which is used in this alignment, and it's here. And if we go for Ctrl A and Ctrl C and take it to gene view and do a paste here, Ctrl V, do I want to add this alignment of 25 sequences, which is in the clipboard? Confirm I did want it, and there we go. This is super highly conserved, so it seems there are hardly some very few differences there. And then some sequences that have odd looking insertions, they could be retained introns, which are problems in the gene prediction, or maybe they are real insertions, hard to tell. But anyway, the sequences were technically very simple to get there. I take a new blank window and close the previous one. Then let's have a look at the gene trees. So while we are on the gene page, we have all these comparative genomics tools. We don't find them on transcript page, so if you are lost, so just check that you have the gene tab open. So clicking on gene tree, it takes a while to load, so I have preloaded it here. And now, in addition to the current gene tree, we also get a list of the supertree of related gene trees. So presumably this includes a number of paralogs which are with large numbers of genes, and then it has some kind of uncertain gene trees, outliers which are not grouping well into any of these gene trees. You can see these trees containing just 5, 3, or 2 genes. And let's look at the gene tree here. So the red color here is indicating our focus gene, and if there are any blue lines here, it would indicate that we have paralogs included in the gene tree. And here the single sequences which are opened, like the human and gorilla version, they are shown in light green, and they show where in the alignment of all sequences these proteins have some sequence. And these small black tick marks here, or small black lines, indicate the exon boundaries. So seeing these exon boundaries at the region of these white parts makes us think that maybe the alignment was actually showing some introns that have an open reading frame which shouldn't have been predicted there, but I'm digressing. And then when we have these triangles, they indicate that we have a compressed part of the tree. So in this case we would have 30 homologs, and these dark green things have a different meaning. So for single sequences we have the aligned sequence and no sequence, but here dark green indicates that we have at least 2/3 of the data supporting that part of the alignment. And again, it looks very similar in pattern, so we might think that the exon pattern of this protein is pretty well known. But let's open the tree. We'll go for expanding the full tree. We have the link here in the bottom, View Fully Expanded Tree. This will open every branch in the tree and show the single sequences. And now you can see from the names that these are all HDAC3 genes. And now this background color indicates the group of animals we have there. For instance, we have rodents here with pink background, and here we have birds in yellowish background, and around birds we have reptiles split in two, so that we have turtles and tortoises here closer to the birds and lizards here. And if we click on the node here, we can make sure that it's actually lizards, and there's the estimated time of divergence since that node here. And if we want the sequences, we have exporting subtree here, and this would give us the alignment of just those sequences. We want to have it in FASTA format, but we could also get some different tree formats if we only want the tree, not the sequences. But the alignment in FASTA should do us the trick. And going for download, again we can choose between saving file or opening with some program. That depends on your computer how you have set it up, but here we have it. And Control A, Control C, and a quick check in C view, Control V, 7 sequences, and here they are. Again with some insertions, but not as many as in the example of the previous ortholog table sequences. This is all I want to show of gene trees. It's a lovely tool, a very complex one if you want to find out exactly how they are built, but even without the full understanding, you can make use of the nodes which work as links and use them for just studying the gene tree, seeing which sequences are complete or incomplete based on the green alignment symbols, and then retrieving alignments of just some subtree in your gene tree.